In the last episode, we saw how the virion that entered through endocytosis was degraded in the endosome and receptors were activated that induced an antiviral response. But at the same time that this happened, another virion introduced its genome into the cell by membrane fusion. As it is not connected to a motor protein, it is almost immobile in the cytoplasm of the cell. But entering the cytoplasm is not enough to infect it. The cell also has antiviral defenses in its cytoplasm that the virus must overcome in order to take control of the cell. The nucleocapsid proteins that are covering the viral genome begin to randomly detach themselves, leaving the genome naked. As it is not attached to the nucleocapsids, the genome begins to fold, forming loops that are essential to regulate its translation to proteins. We call this structure secondary structure of RNA. But to start synthesizing viral proteins, the genome has to randomly meet ribosomes. The ribosome is a complex of proteins and RNA so big that it stays almost immobile in the cytoplasm. So it is much more likely to meet with other smaller proteins like ribonucleases. Ribonucleases are enzymes that break down RNA that is damaged or must be discarded. Ribonucleases XRN1 degraded from the front in the 5' part, the dis3L2 degraded from the back in its 3' part. Cell messenger RNAs are not immediately degraded by ribonucleases. Messenger RNAs contain a molecule that we call cap in its 5' part, and this prevents the XRN1 from binding and degrading the RNA. In the 3' part, the messenger RNAs contain a series of adenines that also prevents dis3L2 from binding and degrading the RNA. Surprisingly, the coronavirus has evolved the ability to synthesize a false cap similar enough to evade the binding of ribonuclease XRN1 and not be degraded. The only difference is in a methyl group that is missing from the viral RNA cap. On its 3' side it also contains a series of adenines that prevent the binding of the ribonuclease dis3L2. The viral genome has managed to evade the first intracellular defense. But he still needs to evade more aggressive defenses. The RIG and MDA5 proteins are very similar proteins that detect RNA of viral origin and are as important as toll-like receptors. The RIG protein detects false capped viral RNA, and MDA detects very long regions of double-stranded RNA. When either of the two is activated, the antiviral response and degradation of the viral genome are initiated. But as we have already seen, the viral cap is too similar to the true cap, and this similarity decreases the affinity with the RIG protein. Even so, some RIG proteins manage to bind to the false cap, and the RIG protein attach themselves to the viral RNA. When 4 RIG proteins bind to the RNA, they form a tetramer. Then the TRIM protein adds ubiquitins to stabilize the tetramer so it can bind to IPS1 proteins. But the virion has a weapon to counteract the RIG proteins. The nucleocapsid protein that was introduced to the cell along with the genome is what we call the Swiss Army knife protein. Because it not only fulfills the function of packaging and protecting the genome of chemical damage, it also has several very important functions at different stages of the viral cycle. One of them is to interrupt the antiviral response, giving the genome more time to take control of the cell without being detected by the cell. This is achieved by binding to the TRIM25 protein, inhibiting its function and preventing it from adding ubiquitin to RIGs, blocking the activation of the antiviral metabolic pathway. But the cell is not so easy to tame. It contains another similar protein that is very effective against coronaviruses, MDA5. This binds to regions of double-stranded RNA and forms a ring that surrounds the RNA. More and more MDA5 proteins fold to the RNA to form cylinders. Every 4 MDA5 join to form ring-shaped tetramers. These rings serve as primers that initiate the polymerization of filaments in the membrane of mitochondria. Transmembrane IPS1 proteins found in the mitochondria bind to the ring of the MDA5 protein tetramer, and more IPS IPS-1 proteins begin to bind, forming a polymer of IPS-1 proteins that spreads on the surfaces of mitochondria. These filamentous complexes serve as a platform for the TRAF3 and TRAF6 proteins to bind. And as we saw in the last episode, the TRAF3 and TRAF6 proteins serve as platforms where other proteins join to be activated, and these in turn activate others, and thus begins the signaling cascade that culminates in the activation of regulatory proteins such as IRF3, IRF7, nuclear factor kappa beta, P38 and JNK. The nuclear factor kappa beta is removed from its inhibitory part through its degradation in the proteasome. All these regulators are imported into the nucleus to activate antiviral genes. Together, the innate defenses of the mucosa, endosome, and cytoplasm manage to destroy the virions that try to infect the epithelial cells of the nose. But we have not seen what happens within the nucleus of the cell and what genes are activated and what proteins are generated from these genes. In the next episode, we will see alpha and beta interferons and their effects on cell metabolism.